I mean, the body is designed to be a broadband absorber or broadband user of the broadband thermal emitter, the sun, moonlight. Whether you look at even the thermal vents down in the ocean are broadband thermal emitters, and fire. We love to sit by a campfire. It's huge amount of near infrared, our infrared and near infrared relative to the visible, and it has effect. It makes you more sleepy, whatever. But I think that, and I'm not being negative towards the PBM people. I mean, I think that if it makes you feel better, that's good. My concern is that it may be helping one thing and hurting another, because the body does assumes that it always has all the wavelengths, not just a very narrow band of it.
Hello and welcome to the channel. I'm Sarah, and today's guest is Scott Zimmerman, back for the third time. In this conversation, Scott and I discuss light and how it affects our biology, water inside the body, hormones, metabolic functions, mitochondria and health, and more. Scott explains how data surrounding light, especially blue light and near-infrared light, has been misinterpreted and what this means for us and our health. Scott very kindly does a presentation to walk us through some important aspects of optics, such as biological antennae, refractive index, measuring light, the role of water in light absorption. And this helps us to understand circadian rhythms and metabolic functions, but looking at it through a light and an optics lens. Scott's an optics expert and he holds over 50 patents. And in this conversation, because it's our third one, there's a few times where I interrupt Scott to ask him to explain things in more simple terms, and he's very kindly obliges. If you find that the level here is something that's a bit above you, but you're still very interested in quantum biology, I've just made a brand new course that's $19, and it's designed for people who are new to quantum biology. It's the Quantum Biology Starter Kit, Energy and Mitochondria from the Ground Up. And it comes with the handouts. There are 50 handouts or slides, and also it's a recording so that you can watch a video as well as read if you want to get up to speed and understand quantum biology. In this course I cover light, water, magnetism, charge, deuterium, and it's a great starting place for beginners. Welcome back to the channel, Scott, for the third time. So today we've got a treat of some optics lessons of how it's relevant to biology and mitochondria, and also some interesting data. Some of it's really old and some of it is all brand new. So I'll just hand you over to Scott now.
Thank you, Sarah. Yeah, I just wanted to appreciate the opportunity to go in and talk a little bit about what we've been doing lately. I've been working with Glenn Jeffrey and Bob Fosbury quite a bit, and Roger Spout. And what we're finding is really I believe, some kind of fundamental new news, I guess you'd call it. But first I wanted to kind of explain what's going on as far as we're trying to present data that is shown in a way that is more applicable to various things. So what I got up here on the screen are two graphs. They're actually identical. They're both the amount of energy reaching the surface from the sun at high noon or whatever you want to call it. But they have very different effects on people and how they view what we're doing. The lighting industry uses the graph on the left, which is in watts per meter squared nanometer, and as a function of wavelength. And if you look at that graph, your first impression there, most people's first impression is, well, the visible is much more important than any of the other wavelengths. And in fact, that was used in great extent for the justification that, well, all that's wasted energy. That's just heat. It's not doing anything. Therefore, we should get rid of it. And that's exactly what the industry did. We took and got rid of all the infrared component, 90%, which represented 9% of the spectrum. And you said, okay, we're going to put all that energy into the visible. And so what we ended up with is unfortunately, what we didn't realize or didn't consider is that in nature, we're never exposed to narrowband emitters like we have with LEDs or OLEDs. We're always exposed to what we call broadband thermal emitters. And those extend from 250 in sunlight is 250 nanometers out beyond 6,000 nanometers. Now, like I say, the graph on the left says, there's not much going on. But if you go to a different industry and you plot exactly the same thing, the number of photons hitting the Earth's surface, but in units of measure that the solar cell manufacturers industry uses, you get the graph on the right. Now, the gray is all this same gray as far as what it represents the infrared. But the blues and the greens and the yellows and the reds are seem less significant. And the reality is they are less significant because what you're looking at in a solar cell using the units of the solar cell is what the mitochondria cares about, what our metabolic processes care about, because that's where are the energy levels that are most effective, are most similar to the activation energies that we find in biology. So again, I'm not trying to make it into more than it is, other than just to say how you present data. And Sara, we had talked earlier, was talking about some of her stuff. How data is presented tends to have a big impact on what you think is the relative importance. But if you really care about all the metabolic diseases and the things we're struggling with the mitochondria problems we're having, then it's actually the graph on the right that has more relevance and is more illustrative about what's going on.
First of all, for people who are not familiar with optics, if you could explain to them why using watts per meter squared is misleading, and because the other axis is photon flux, and that's represented, I think, in electron volts. So I think that's kind of really important, that we're distinguishing between just raw power output. And now it's a bit more specific now if we're talking about electron volts.
Yeah, the graph on the left is doing everything in terms of, photons per second per meter for area per unit wavelength. So the graph on the left is looking at things their wavelength, which not surprisingly, if you're talking about visual or eye response, then wavelength is a reasonable metric. That's a reasonable metric. But when you get into trying to look at metabolic, it's all about how many electrons can we move on the electron transport chain. Okay. EV is a unit of measure of energy. Okay. And if you look at the electron chain, there's all these different stages breaking down the amount of activation energy that you need in order to overcome the barrier to get to the next point, kick some more protons up and run the gears, essentially. If you have, it's called photon-assisted quantum tunneling. If you have the energy being provided by various photons into the body, those photons can assist or make the ability of that electron to jump that barrier occur more effectively. And you talk about that in terms of, and so when the solar cell industry, which this is from the solar cell industry, it's not my graph, is basically saying, okay, if we had a band gap that lined up with the 1 EV, say, for an example, a material that would actually do it, then we could get an electron out of that. And they're only interested in how many electrons. Well, the body's really only interested in how many electrons make it over the barrier, because the quicker and the better that they can go over those barriers, the faster the rotor will turn, the more efficient the process will be, the less reactive oxygen. The problem is that with the higher energy photons, which are the reds, the yellows, the greens, and the blues, they have so much energy, it's coming in like with a sledgehammer and knocking it in, and the excess energy goes into reactive oxygen species, is what our point being. And that then can lead to damage, can lead to less efficiency because the body has to deal with the extra reactive oxygen species. So that's our point is that, and what's miraculous about this is if you move down to where Bob's showing the same graph, taking the right and just expanding it out a little bit. The green is the green little lines are the same as what's over on the right graph that's filled in with gray. But there's this unique phenomenon that only astronomers really know, because I'm sitting here talking to Bob and I say, hey, Bob, the solar industry's got this peak over here at 0.75 EV. Now, 0.75 EV in wavelength is essentially 1660 nanometers. So it's deep into what we call more the shortwave infrared. And Bob said, yeah, of course. That's the hydrogen minus ion opacity window. I said, huh? And, the bottom line is that, for astronomers, this is a well-known phenomenon. What happens is that sunlight or the sun's atmosphere is essentially got a window where wavelengths that are deeper down in the body of the sun are allowed to escape in this particular wavelength range. Now, it just happens, and you can call it coincidence. I call it what actually is probably one of the coolest phenomena, is that life on Earth in general operates using an average activation energy around 0.66 EV. So it's like what I believe what's going on is that this little extra bump was used as essentially a primer for us developing a lot of why the electron transport chain has X number of stages, why it needs how much activation energy. And it's just an amazing coincidence that we have this sharp peak that is part of the sun that comes all the way through down to Earth. And I believe that what we're showing is something that is fundamental to why we're built the way we're built. And you can tie this in with quantum tunneling effects as far as the time constants. There's a number of different things.
That makes sense because the invisible light clearly was used long before the visible light came along because things didn't have eyes.
Exactly. And, when you're looking at it, there's a lot of life forms that don't have eyes. They're not, but they do have surface area. And what is really cool if you look at the pictures down below, in the region that this is occurring, all life forms are basically reached their maximum absorption. It doesn't matter what skin color you are. In this wavelength range, you have black skin because it's all about the water that's doing all the absorption. So water is absorbing strongly. And the argument being put forth, especially by the Guy Foundation, some of the people in the Guy Foundation, is that water is the chromophore. And that if you look at what happens when a photon enters a mass of water, it can get absorbed very quick. It affects all the other water molecules that are kind of in this pool. It's kind of like throwing a rock into a pond and seeing the ripple effect. You have to start thinking about this as being three-dimensional and a function of time because you've got all these photons coming in, essentially hitting this water matrix and changing the relative distribution of molecular activity, which then translates into how quickly or how easily it is it's no different in a lot of ways than heating up a reaction in a beaker.
But also, I think with water, first of all, all the molecules are just doing their own thing. Some are structured, some are not. And as soon as you start to add light in on top, the level of complexity just explodes through the ceiling.
Yeah, you're exactly right. And you got other things that are happening where, in the same region, some of these wavelengths are generating hydrogen peroxide, or other things. And that's coming into play in the whole thing.
But actually, would you be able to talk about the wavelengths that water does absorb well in? And I know other electromagnetism and waves interact with water, but then I think what you said about a different higher energy light changing the chemical properties of water. So that's something I think is really interesting.
Well, I mean, if you look at the picture on the right of the young lady, that's in the UV regime. She has a darker skin because water is strongly absorbing in some of the UV. But, as far as liquid water, you have to also keep in mind that there's liquid water and there's vapor water, and they have very different characteristics optically. Those little windows or gray lines that you're seeing in the graph up above on the right were associated with going through the atmosphere, which is water vapor. And so there are windows. The astronomers use these windows to be able to look at stars that they can't see because of the water vapor absorption in other wavelength ranges. So it's a really fairly complicated arrangement. But in general, the shorter wavelengths in the UV absorb strongly in the water. That's how we get, and they have enough energy where they can break bonds. The graph on the far left is water being absorbed again, but the the energy is being imparted in a much more gentle way, I guess I'd say. There's more photons. There's actually more photons per volume being generated in the graph on the left than it is on the right, because while the UV photons have high energy, there's not very many of them relative to what's going on over in the infrared, where they're lower energy photons, but there's a lot more of them and they're getting absorbed in a very small volume. So the highest photon density in the body when you're in sunlight is actually out here exactly in the same area where most of the biological processes are operating or have activation energies in that range.
Because I'm not an optics person, but the way I think about that is to do with charge distribution, that just because you've got more charge, it doesn't mean it's better, because if you can't distribute it properly, then it's no good biologically. So is that correct about how
Yeah, I think that's a good analogy of what's going on. I mean, you have all these water molecules, and there's some really great work by a gentleman, Nathan, I forget his last name, out of the Guy Foundation. He just gave a talk about it. And he was modeling the effect of water molecules singly, but also in groups. And what you find is that there's various reconfigurations that occur within the water molecule, and you can literally transfer electrons using those changes in configuration very rapidly. And then the more water molecules you add to the model, you have these longer range effects. One of the big problems with a lot of photochemistry is that, you get a photon to come in and do something, hits it and breaks it into a reactive oxygen species. But that reactive oxygen species only has a half-life of nanoseconds. So it can't go very far. It doesn't, the diffusion distance is essentially zero. So there's always been this quandary of how do you get this long range signaling and things of that nature going on? Well, it appears that there are, on a quantum level, there are all these different ways that reconfiguring molecules or their configurations or their orientation or whatever you want to call it ends up being a mechanism for spreading. Like I say, throwing a rock into a pond and seeing the ripple effects out in 3 dimensions. So,
but that's really interesting, because with a lot of this, I was going to ask you later, how does it actually do it? Because with biochemistry, I just think of it as all being local. A molecule has to be next door. Whereas anything that's action at a distance, or people want to call it entanglement or whatever, it requires electromagnetism. But then also, I think what's really important is the way in which water plays a role in this, because we're going to be talking about sort of light bending and refractive index later. I just wanted to ask about the crystalline state in the body and whether that's significant in terms of what we're talking about, or is it just something we don't know yet?
Anyway, bottom line is this is a TEM at the limit, basically, of what we can actually do TEM, and of a leaf, chloroplast. And those periodic structures, I'm an optics guy. What's extremely humbling about this whole process we're going through is you find out that nature for millions of years are building structures that we can't even think about making ourselves right now. But we can start to see what they're doing. And like I say, this is probably one of the most dramatic ones, is that anytime you generate a refractive index, water has a refractive index of 1.3 at certain wavelengths, and the lipids or the cellulose can go up to 1.5. Well, what is that saying? That's saying that you can generate, if you can make microstructures or nanostructures, you can generate selective filters. And that's what's going on in this particular case. What you're looking at is in a chloroplast is a structure that's that the plants generate. And what happens is that try and say this the right way. The photons coming into that structure, some are accepted, some are rejected. They act like you've ever seen a dichroic mirror that has different colors. And what they're doing is they're building layers upon layers of structure on the surface of that glass. And what it does is some photons can come in and be reflected back out. Some photons can be transmitted. But what happens is that not in that process, each one of those little layers, the higher index one, the photons actually have a higher probability of being in that little high index layer. Well, that's exactly where plants put their chlorophyll. And what it makes is this amazing process where light comes down through, that hits the leaf, sees this structure. The green is rejected because the majority of the green is rejected because it's of no use, because chlorophyll has very narrow absorption bands in the reds and the blues. And instead, the red and the blue photons are actually localized right where the chlorophyll is. And then in the space between is where the oxygen is released, which helps generate a higher refractive index difference, because the more closer it comes to air, the more closer it comes to refractive index 1. So you get this enhancement effect based on structure. And you see this in jade leaves, you see it in diatoms in the ocean, you'll see all these periodic structures. All those periodic structures do is localize photons in a particular spatial place or wavelength range.
But that's really important, because everything on a quantum level is tiny signals. So you can't possibly just have the light randomly going wherever it wants, because there'd just be a huge amount of noise. So what you're describing it's the method in which biology propagates or makes the signal using, we would call it dichroic mirrors, or there's all the different physics terms that we could use. But I think it's really profound, this idea of the structures and how it's not just the light going anywhere, the way in which the body funnels it about, almost photon by photon.
And, it's just the level of adaptation and brilliance of the design is evident when you start thinking about it from the standpoint of there's only about 20% of the surface area in a lot of cells covered with these little chloroplasts that contain these little microstructures. They tend to be oriented. Whenever you take a picture of them, they're oriented in the same direction, because I believe that they're actually rotating to, like a sunflower in the sunflower field rotates. These little pups can rotate to maximize the efficiency of getting the reds and the blues in the particular bands where they can be most effective. But what's really cool is that it also transmits a lot of it only takes out a little slice, a little portion, but it lets the rest go through to the leaf down below. It takes a little portion, goes down to the next leaf down below. It takes a portion. So it's got, you look at a tree and you see all these leaves, the way they're arranged, they're actually optimized around the idea that I'm going to only take what I need. I'm going to very efficiently extract just the reds and the blues, and I'm going to let the rest, everything else, some high percentage, go on down to the next leaf. And by the time it's all said and done, it's very effectively stripped out only those wavelengths that are most important. So at the end of the day, it is the most efficient. Bearing in mind that plants are 200 million years more advanced than we are, it's pretty interesting that they've gotten to this level of complexity. And I believe the reason it's that's so complex is because what matters to the plant is extracting those wavelengths that are most important to it with the least amount of volume necessary so that everything else in the cell can function and do its thing. The mitochondria, all the other functions that are going on in plants. But it is an extremely optically elegant design. And what you'll see is that as we get better at TEM and our ability to see deeper and deeper into structure or into the cells, without screwing it up. 99% of the mitochondria pictures that you see on the web or in the literature are damaged mitochondria. It's really hard to not mess with the structure when you're fixating the sample for doing TE.
I did lots of electron microscopy, and lots of structural biologists actually think it's an artifact because you have to dry it in epoxy resin and then you put osmium in it, and it's one of these things, it's still nobody's thought of anything better yet. Although I did actually meet a Scottish man who's got a patent for its live cell imaging, like almost like tomography, because it's exactly this problem that once you fix something and freeze it, that's not how it behaves in nature. So how do you know you're not making artifacts by accident and then basing hypotheses on an artifact?
Yeah, and that's what's cool about this is that this is from Brookhoff, the pictures you're seeing. What he did is then used a Bragg grading software package to estimate what the reflectance characteristics would be. And lo and behold, given the index of refraction that we have that the plant has available to it, he was able to show that green was rejected. And right where the red and the green, there's 2 red and 2 blue chlorophylls. Right at those wavelengths, that structure has its minimum reflectivity. So it maximizes, and they're deep spikes that just line up perfectly with the peaks of the chlorophyll. So,
Would you be able to explain what refractive index is to somebody who doesn't know at all what it means and how it's relevant to biology?
Well, whenever you enter a material, you're changing the relative, you have to adjust the wavelength and the velocity, or the wavelength, let's see, which was it? Wavelength stays the same, velocity changes. You essentially slow down the photon. The minute you do that, you're introducing a phase shift. And then if you put that, you have another wave coming in with another phase shift, you can have interference effects. So these are basically just, not exactly, but when you have a moire pattern that you see on 2 screens that are blocking, you can essentially have different ways, I guess. I don't know a good way to explain it.
I suppose, doesn't it, one way it bends the light. So that's one way of altering the signal direction. But also if you slow down something, then you're modulating. It's like a sort of a volume switch in some ways. So it's just part of the thing is we don't understand at all the fine tuning of what goes on in the body's optics, but it's just something that a lot of people don't really appreciate or understand what refractive index is. But would you just say in simple terms, it's like some kind of switch or like a modulator for direction change? And
well, I guess no, I really think there's a change in speed because in order to if we enter material, it's essentially slowing down the photon rate, I guess would be a better way to put it. I don't know a good way to put it.
Maybe it's like a time, because if you slow something down, you've got a time sort of differential in there.
Well, we typically talk about phase delays associated with, you have a wave coming in. And the problem is this is a wave. We want to talk about photons. They're not a wave, but they are a wave. So I guess I'd have to acquiesce to somebody more smart than me how to explain that in an easier term, other than, when you have a photon coming into a material, and depending on the density of the material and the wavelength that you're dealing with, you will have a slowing down associated with it, and that changes the phase. And then based on that, its interaction with other waves are going to be altered. And if you do that with enough periods, then you can create structures like this. And, the water in there is 1.3 and the lipids are about 1.48, 1.45 as far as the refractive index is concerned. That gives you, when you put those all together, you end up with structures that can, reflect certain wavelengths of light and transmit other wavelengths of light.
Also, will what's dissolved in the water change? We're obviously going to change the water's refractive index as well, so it gets ridiculously complicated quite quickly.
Yeah, and like I say, what appears to be going on in the structure is that it's generally, you have to start, unlike other processes, biologically, what's it do? It's doing something. It's actually generating oxygen. And so where's that oxygen going? Well, it's going into the low index water layers, and that is lowering its effective refractive index slightly, which enhances the contrast, because at the end of the day, any of these structures There's two functions, the number of periods, the shape of the periods, and the refractive index contrast between the different layers. And low index contrast like this, in your dichroic mirrors, they'll have glass and they'll have aluminum oxide. Big difference. In these little structures, there's a very slight difference, but it makes it so that you have extremely efficient effects, if you get enough periods built into it.
I want to thank my sponsor, Lightwater Scientific, who helped me deliver free, valuable information to you and also allow me to have great guests on this channel. Lightwater Scientific produce the purest and most deuterium-depleted water in the world. Depleting or lowering your deuterium has numerous health benefits, from better metabolic health, more energy, greater resilience, less brain fog, increased physical performance, enhanced healing, and healthier mitochondria. Deuterium is an isotope of hydrogen, also known as heavy water, because it's bigger and heavier than regular hydrogen. Even though deuterium makes up only 0.02% of hydrogen, it's found everywhere. Deuterium is a burden to your body because it interferes with the intricate chemical reactions that occur by slowing them down. Deuterium is kryptonite to your mitochondria, and we need healthy mitochondria to produce energy. There's over 60 years of scientific research on the negative effects of too much deuterium on health and also the positive benefits of lowering deuterium in the body. Lightwater deuterium-depleted water is 100% safe and non-toxic. In fact, it's the closest in structure to the metabolic water that your mitochondria produce. Lightwater deuterium-depleted water comes in 5 or 10 parts per million glass or plastic bottles, so level up your health by leveling down your deuterium today. The purchase link is in the show notes, and use code Dr. Sarah for a discount off your order.
Oh, no, it does. It's like biological optics and optics we build have the same principles, but then biology has its own special nuances, which we're just learning about.
That, I think, is what's been missing in a lot of ways from not only biology, but quantum biology in particular. I would argue, as you just said earlier, once you get the structures, then you can start having quantum effects be more relevant, I guess.
Absolutely, because it all depends. It's all back to the biochemistry is all local and action at a distance. It has to be what we're talking about now, the optics, the water, the electromagnetism. But yeah, I think we've kind of gone a bit too nerdy. So I can't remember what we have next for the slide.
Well, just one more nerdy thing, because everybody loves biophotons and all this kind of stuff. But I would argue very strongly that if we do get into the world of biophotons, the structures around it, it's kind of like I used to design antennas. And, if you look at an antenna array, you can get what they call a Bragg grading enhanced antenna. And what it does is if biophotons are going to be communicating cells to cells and all this, the ability to form a structure that shrinks the wavelength range, looks at directionality, things of that nature, is probably going to be what we find, because the reality is that our body's got all this noise associated with all the photons coming in from all different directions. But biophotons would have to have some kind of an antenna system in order to be able to extract it in the same way you extract a signal via spread spectrum or whatever in our modern cellular communications. So structure is going to be, I think, one of the key elements to getting a better understanding of what's going on at the quantum level and in general in biology, because virtually every thing you look at, birds' wings, butterfly wings, diatoms, everything has structure of some type. And most likely that structure is being used in some way by the body that we don't quite understand yet.
That's really important about the antenna. So basically, sometimes people have heard the word antenna, but they don't actually appreciate the context. So it's back to our conversation about how in quantum biology, there's very small signals and potentially lots of noise. So you need an antenna that's able to sort of pick out the photons or the signal that you want, a bit like it's just a ham radio can pick out certain frequencies out of all of the rest. So in terms of what you'd say something is an antenna biologically, what would you say in the body are antenna? inside the cell.
Well,
Because water can be an antenna, can't it?
well, but again, I'd say it's structure that really matters. If you look at the reticulum around the nucleus, it is very highly periodic, has a high level periodicity. If you look at DNA itself, it has,
Oh, yeah.
the first GPS antennas were double helixes. There's all kinds of games that we don't understand that we could play here with our structure. But at the heart of it, no matter what it is, as a function of wavelength, the introduction of periodic structures or antenna structures seems to be well within the range of anything that the nature is doing. And this is just one classic example of how, I didn't make this up. This is what occurs in the leaf. And you think of how many of those little structures are being generated in all the plants across the planet, and the effect it's basically making it so it only extracts the portion of the spectrum that it needs to be efficient at generating photosynthesis and no more. In the longer wavelengths, plants, as we've shown, the actual cell structure on the outside of the actual cells scatter light in the near infrared very brightly, almost 100%. So, and why are they rejecting it? Because it's not useful, because photosynthesis has a very limited range of temperatures where it operates efficiently. So it is optimized around getting just that portion of the spectrum that it needs, and taking out the greens and the infrareds or near infrareds and kicking them out.
But also, biology doesn't like wasting things. So just because something's, we said, rejected, it might be reflected, but then used for a different process or for signaling, because that's the thing. There's a whole physics behind when something bounces off a reflective surface, and biology is very conservative about how it uses energy. So I'd imagine there's something in what gets reflected as well as what gets gathered.
Well, and I would argue that's been at the heart of what we've been talking about the near infrared and the infrared. Plants reject. We walk down through a forest. We have a pair of sunglasses on from the trees where they're absorbing certain wavelengths, but they're transmitting and reflecting other wavelengths, so that our entire body is bathed in these near infrared photons that I would, that we are arguing have a huge impact on the efficiency of mitochondrial production of ATP. It also seems to have an impact. I mean, all the photobiomodulation people are doing reductionist type approaches where they're looking at a particular wavelength and saying, hey, look, the wound heals faster. I could just as easily have gone to 850. I could have gone to 1064. It's changing the hemoglobin levels in the bloodstream. It's changing, affecting how people think. We're all looking at all these little micro points in a much bigger design, I guess. I mean, bear in mind, the body is being exposed to all the wavelengths when we're outdoors at the same time, and all the processes, depending on what wavelength you're at or what, are being affected at the same time. It's all simultaneous. When we run experiments, we go and we put 670 nanometers on here, and we see this great improvement in glucose. Yeah. What else did you do? Because if you had 850 over there, would have been better. If you had 1064, would have been less. If you had 6,000 nanometers, what would be the effect? And that's what I think is really coming out of this work is that the body is using all these different wavelengths. They all are interacting with various different parts of the metabolic and neurological and all that other stuff. I mean, before I got doing this,
this ties back to what you said right at the beginning, that too much of a given light, too much energy causes free radical generation, or we know with optics you can photo bleach and damage everything. So I completely agree with you about the photobiomodulation and individual wavelengths. In some ways, it's just way too much energy. And as you said before, how do you know what else you're making and creating? Because the body isn't used to receiving all that energy all at once.
Yeah, I mean, the body is designed to be a broadband absorber or broadband user of the broadband thermal emitter, the sun, moonlight. Whether you look at even the thermal vents down in the ocean are broadband thermal emitters, and, fire. We love to sit by a campfire. It's huge amount of near infrared, our infrared and near infrared relative to the visible. And it has effect. It makes you more sleepy, whatever. But, I think that, and I'm not being negative towards the PBM people. I mean, I think that if it makes you feel better, that's good. My concern is that it may be helping one thing and hurting another, because the body assumes that it always has all the wavelengths, not just a very narrow band of. And, we got into trouble with blue light. Everybody's down on blue light because it's a spike. Well, what do you think 670 is or 850 is? It's another spike. You're essentially only exciting a very narrow range of wavelengths, and and in order to get an effect, you want to do it quickly. So what's happening? The PBM people modulate up to the intensity level, and before long you get somebody who gets hurt because you don't have especially in the near infrared. My big pet peeve with the industry is that people don't have the ability to look away from 850 nanometers.
Yeah, because you can't see it. But also, I think it's something, I would just call it's science in its infancy, and people have jumped ahead. And normally, if that was a drug, it'd have to go through clinical trials, whereas none of these lights have gone through any kind of clinical trials, and light is a drug. So that's my personal concern. I don't think there's anything wrong with it. I think it's fascinating. I just think we've underestimated it, and it should be, light is a drug, it should be treated like other drugs are treated in science and tested properly.
I agree. I mean, I think you're 100% right. The problem is that, that's why we do what we do with our light sources, is we put visible LEDs in with incandescent systems, and then we try and balance that out to get the best mix that runs from 400 nanometers out to beyond 3,000, out to 6,000. But to be honest, it cost us a bloody fortune to actually even measure out to 3,000 nanometers for our light sources, because everybody's running around nowadays with these spectrometers that only go out to maybe 700, 900 nanometers, totally ignoring the fact that that's not where the real action's going on, in my opinion. You highlighted
it before. Everybody's obsessed with the visible spectrum, and they act like the rest of it doesn't exist.
Well, yeah. I mean, I've had people measure our light sources and say, there's nothing going on out here in the near infrared. Did you measure all the way out to where we can talk about the next graph will kind of explain why we're doing what we're doing.
Yeah.
But so anyway, bottom line is that if you put the solar spectrum that we receive on Earth in terms of photons per second, meter squared, electron volt.
Okay, so here we're looking at wavelength, so we'll just pretend that will be color. I know that's not the right way to describe it. We're looking I actually wrote a Substack on that. But anyway, so we're looking at wavelength or color on the X, and then on the Y, we're looking at spectral irradiance again, but this time it's electron volts.
Yeah. And the red line that's on this graph that you see below the data is what most people do with sunlight. They approximate it by a black body emitter, follows Planck's function. You can plot it, it has a color temperature. Yeah. Okay. So that's what you would expect. But this effect that is occurring based on this opacity window in the sun's spectrum gives you this bump up. And it just is too much of a coincidence, in my opinion, that the activation, average, life activation potential, or whatever your activation energy of most biological processes fall in very close to that peak. And, we're talking about the difference between, those life forms who took advantage of that peak. That peak has been there for billions of years and will be there for another billion years, probably, barring an explosion.
But, so we're looking at we've got the 0.75 electron volts peak. So that's the solar. And then we've got the 0.66, which is the average. Yeah. So what's
for all terrestrial life forms?
Right. Okay.
Now, the only ones that don't have that high of an average. So these are essentially the average activation energies necessary for the process of the reaction to occur. So biologically, we divided up the electron transport chain into multiple stages so that you could get X number of protons out of an electron, that was generated. That electron was generated can be generated using photons. In fact, it's a fairly well-known process in this laser or the physics world. It's called photon-assisted quantum tunneling devices. Make them. Well, the body's been making them for several 100 million years.
So is this graph effectively saying that the near infrared, it drives life, but that makes perfect sense because otherwise we die when it's dark?
Yeah, I mean, you don't instantly die. I mean, if I were to draw another graph, it would be the Maxwellian distribution coming off of our surrounding temperatures. And it comes up to about a half an EV type range. So you've got two things going on in nature. We are designed that we can survive in the dark.
Exactly.
We use enzymes, we use proteins to overcome the activation energies, necessary barriers to get through life. Okay. But it does so using very low energy photons and energy levels and various structures, like I say, enzymes and proteins. So we can survive for a while. But the minute we walk outside, all of a sudden now we have this additional radiative input that just happens to kind of overlap they kind of overlap in this region where the photon energies that are available can assist or enhance our ability to operate. And especially people with, now that if you're looking at everybody being the same, we all have very strong absorption in this region. We all can take advantage of this process. And when you're outside, you feel a little bit better. You feel a little bit, typically, you'll feel a little bit more energized versus you're sitting in a dark hole. I mean, you look at some of the astronauts coming back from the space station, how they look before and after. They look awful, or the hostages that stayed in a tunnel for a year. They look really bad.
But that's the thing. That's because they've had too much dark and not enough light. But just as a question to you, with this idea that we can't live without near infrared, because this graph pretty much shows it, do you think some of the problems with artificial light at night is not just the blue light being there, it's people thumping about during the night when they should be sleeping, either working or doing stuff in the house, and because they don't have the near infrared because it got taken out of the LED, do you think that's part of the problem of why the artificial light at night is so bad? Not just because it's blue light and people are exposing themselves to light when they shouldn't be, but it's also the lack of the near infrared.
I totally agree with you. And just I'll run down here just to show you this one graph.
Because it is really important, that graph. I didn't point out something else, but I think it's something that, like you say, we keep focusing. There's lots of different ways to look at the same data and look back to what we know about, okay, artificial light at night is really bad, but what can you do if people have to be exposed to artificial light at night?
Well, I think the problem is that it's easy to get out of sync, but the issue that I have with artificial light at night is that the idea that it's a blue problem is just wrong. This is a graph of the response, what happens when you hit twilight. The purple line is measured, the light purple one, and the blue one is a model that Bob Fosbury generated. And the minute we saw the graph, it was pretty obvious what's going on. This is a graph. The purple is a graph of what is the light that you're exposed to. If you look, everybody talks about sunset and they say, oh, it's a beautiful red and orange. Yeah, for about 5-10 minutes, it is. But if you look to the left or the right, it's all blue and violet. If you look behind you, it's blue and violet. It turns out, based on the scattering characteristics of the atmosphere, that the more reliable timing element for circadian is blue and violet at night. That's what's turning on and off your circadian response. Because if you look at that I put in the action spectrum for melanopsin, it perfectly aligns with that spike. That spike occurs whether or not it's cloudy or clear. It has to do with the basic scattering characteristics of the atmosphere. So the body is actually looking at the blue characteristics, and you bear in mind, it's not just blue, but it's the intensity level dropping off the blue. But this is the actual key to circadian, in my opinion.
So just to interrupt you again, so when it comes to setting my circadian rhythm, I sometimes get up at daybreak and then I like to go to bed early. So I actually miss the sunset sometimes and the twilight. So do you think there's a lot more in twilight and daybreak than we previously realized? And that's the thing that's setting the clock?
I think so. And purely from a optic standpoint, it's the blues and the purples, whether it's sun coming up in the morning or going up at down at night. But it's because the melanopsin is perfectly aligned with the scatter characteristics of the atmosphere. So I think that I don't know the exact answer to your question. All I know is the circadian, the idea that you can take and turn everything orange or yellow or amber is not really what you're really doing. Is you're cutting down the amount of blue that in some time that may have a good effect or a bad effect, for all I know.
But I think with biology, it's not stupid. It's going to have several checkpoints. So it's going to use the daybreak, and then there are specific signals at sunrise to reaffirm, oh, it's morning, and then there are more later. So I think it's a really interesting concept about what you've brought up about how the blue plays a role in the timekeeping. And I think you were going to say just by bathing yourself in red light in the evening, you're not actually helping anything.
Well, what you're doing is no, I don't think you're actually helping anything, because the bottom line is that there's 9 orders of magnitude going from daybreak or noon to down to twilight. But the only really reliable source of information, or the most reliable source of information, like I say, whether it's sunny or cloudy, is this peak that occurs that lines almost perfectly with melanopsin's absorption characteristics.
I was just going to ask you something else. In terms of optics, what color is the night? If we got rid of all of the artificial light, what color would it be? Because isn't it like a blue, even though we call it black?
Yeah, it is definitely, because there's such a high, all the high angle rays are in play. That's what this graph basically is to scatter off of the atmosphere. So, and you have to think about it. You're not just looking at one point in the sky. You're essentially surrounded. You can look behind you. You can look on the sides. You can do whatever. And it's all going to appear to be kind of a bluish cyan type color.
Yeah, because it's definitely not like dark, as in the absence of illumination. There has to be something going on. I know there's infrared there, but there's also something else.
Well, no, but they're different other graphs. But the bottom line is that we never get to darkness.
Yeah.
I mean, there's always a ton of light. I mean, relative comparison, the biophotons are talking about hundreds of photons per square centimeter. At dark in the night, you're talking about 10 to the 14th photons per square centimeter type numbers. So it's huge amount. Even when it's totally dark, there's still a huge number of photons. And I would argue most of them over most conditions or times of the evening are in the blue and the cyan. So.
Okay, that's fine. Because I think when it comes to making bedrooms pitch black, I wouldn't say that's unnatural, because a lot of humans would have slept in caves that were black. So if a human sleeps in a cave, would you say that that's, I know you can't get like real darkness, but how does that compare to sleeping underneath the stars, being in a cave?
I don't know. I haven't really looked at the numbers, but like I say, I mean, there's a long way. It's kind of like the Feynman's comment about, there's a long way down to the bottom, I think.
Because the thing is, you've opened quite a lot of cans of worms, which is fine, because it's very easy to not question things. And then sometimes I just go back to, okay, people like me making a blackout bedroom, is that artificial? And I think, well, no, because some ancestors of ours would have slept in a cave that was completely dark. Whereas, like we've talked about before, there are benefits if you can to see the real night sky, but it's not beneficial if it's full of artificial blue light.
Yeah, and I guess all I'm saying is that this is an optical effect. It's very clear. When things line up almost perfectly, I tend to believe them a little bit.
Oh no, I'm the same as you.
But I put it forth just as more a thing that I think our circadian theory is a little bit flawed in a couple of different ways, and we can talk about the other way here.
But also, this is what I'd call the elephant in the room, that there's like the obsession with the red light and those kind of colors. And here's a great big elephant in the room. I actually was reading Dr. Lieberman's book on light the other day, and he also pointed out exactly the same as you about the huge amount of blue in the twilight. So it's something, this elephant has been here a while. So it's just, here it is again.
Yeah, and in general, I don't believe much in absolutes. I believe in ratios. And, I think the body, if you're going over 9 orders of magnitude in intensity level, then you need to start looking at things more on a log scale, number one, as far as how we map them. I mean, one of my pet peeves is that we do everything on linear scales because the marketing guys like it much better because they can show how their product's a little bit better than the other. When in reality, eye response, metabolic response, those are all log scale type things. So that should be changing. But ratios, big changes in ratios, be it, light or hormones or whatever, I think is how the body is designed to operate.
Could we go to the next bit? Because I think it might be the melatonin and the cortisol, but you said there was something new, the new data.
Well, I can go down. We can go down there. I want to go back.
I'm just conscious of time and also people's brains because. So I think. Yeah, do you want to? No. I think people might be familiar with this already, but I think you said it was new information.
Well, I mean, I haven't really gone through and published it in a formal sense. What I've been doing is I've been looking at the using sweat biosensors to look at melatonin and cortisol. There's a gentleman that has a company, Cordy, that has a sweat monitor that is sampling every 3 minutes both melatonin, cortisol, IL-6, and TNF alpha.
Is that available commercially? Could we buy?
No, it's not available commercially right yet.
Okay, that's actually a really good. It's like a CGM, like the continuous glucose monitor, but for hormones.
Yeah, exactly. And the point I keep on saying that while I don't disagree that there is a general trend, a baseline trend in cortisol and melatonin, one of the things that comes out is that you have to measure, in order to understand what's really going on, you have to be measuring it, for sampling your whatever biomarker at a fast enough rate to see what's the transient response is.
Exactly.
So every 3 minutes, this sensor goes up. And what I found was really fascinating is that it doesn't discount the standard circadian lore, but on top of that, there are these transient responses. If you do a stressor, and I'll show the one associated with just eating.
Can you just say what the axes are? What have we got on the X and what's on the Y?
In the melatonin, it's picograms.
Is that time? Is it 4 a.m. then like 6?
down on the x-axis is time in day.
Okay, so that's time of day, and then it's just an amount of melatonin.
Yeah, picograms per milliliter. And this is measuring sweat. It's a little biosensor.
When did that person eat? Because it says it's the melatonin graph in response to eating.
Right there is the cortisol response.
Oh, okay. Fine.
So what you see is that my wife and I went out to a really nice Mexican restaurant and had a nice appetizer. I was wearing the monitor. You could see an uptick in the cortisol levels, and then you had the main course. And what's interesting is that what happened is that, and you don't know, it's not like I have the sensor on, but I don't know anything that's reading.
People get really confused at the graphs because the melatonin one it's labeled melatonin response to eating. I just want to check it's the same time on the cortisol graph. Can you just scroll down to the bottom of the,
sure.
Because you said you went out for I think it's it has to be, you said time of day, and then you said that you went out for a Mexican meal.
Yeah, we went out and we sat down to eat at 5:00 in the evening.
Right.
And then we started eating, and what the sensor showed, and the sweat sensor showed, was there was an increase in cortisol, a very rapid increase in cortisol. Then we had the main course even. Once it started getting up to about right there, I don't know if you can see my marker.
Yes.
All of a sudden, there was a spike in the melatonin that occurred, and it went up and then started to come, and then the cortisol started to drop off rapidly.
Okay, that makes sense now, because now I know that the graphs are...
They're lined up.
And also the numbers on the x-axis mean nothing, that we just go by. It's an evening reading, so it's in the evening, and we ate Mexican and the cortisol went up.
They're all in the same line and alignment. You can actually see, even when you go over, we went back and I was in a dark room when I was watching some TV on and off, and you see these spikes of cortisol. Well, if you actually follow that line up, you see a dip in melatonin.
Yeah, exactly. Because I interrupted you. Can we go back to the big peak? Because I accidentally interrupted you when you were talking about the significance of how the melatonin and the cortisol peak are related to each other. Because anybody looking at the graphs will be confused. Well, they won't be now that we've explained it. But do you want to go backtrack a bit to say how the melatonin, the big spike, and the cortisol and the food were related?
Yeah, well, as far as in real time, we're measuring every 3 minutes. What happened was you saw an uptick in cortisol as we started to eat. During the process of an hour-long dinner, the cortisol continued to rise. But then I'd say about halfway through the dinner, all of a sudden, melatonin started to rapidly ramp up. And it ramped up, and then you see the drop-off in the cortisol that was associated with the melatonin spike that occurred.
So when you went out for your Mexican, were you inside, and was it sunset or was it light outside?
It was in the evening. It was a cloudy day, I remember. We just were sitting in a nice booth. It was not very well lit, very heavily lit. So there wasn't anything really.
So it wasn't dark. And what this data is showing is you ate food and it was not after, it was before sunset, and somehow there's a big melatonin spike. Because
what I would, again, I believe in ratios. What I would say is that melatonin obviously affects your circadian, the pineal gland, whatever. But on top of that, as we're going through the day, there are all these transient stressors. You eat something, you do exercise, you make love, you do whatever. When you have those kinds of rapid changes, there is a secondary response that is transient, that where the melatonin is going to put cortisol back in its place, I guess I'd say.
Yeah, that's a good point, that melatonin comes out in the daytime to mop up our messes and stresses that we've made. Yeah,
and if you have the next one is basically the other set of spikes that are there are associated with being indoors in front of a TV that had no near infrared, number one. But it showed that cortisol is a series of spikes. And I guess, what I'm trying to show, and I'm not really trying to show anything more than that, is that there is two levels of control. The pineal gland is obviously not sending the melatonin out, in a spike because of something that I ate. Maybe the gut is, maybe some other thing is, but you can see the same effect on exercise. You can see similar effects. So what is clear is that we are underestimating, or we're missing a significant process where hormones and IL-6, TNF alpha, they're all moving up and down very rapidly, depending on what stressor you have.
But also, from a biochemistry perspective, I think that melatonin made in the brain is not the same melatonin that the mitochondria make, because it's got a different light signature. Even though it looks like the same molecule, I don't think it is exactly the same, but we don't know enough about bioelectronics and biophotonics yet to prove that.
Yeah, but all I'm really saying is that, the idea that cortisol's high in the morning, melatonin's low in the morning, melatonin's high in the evening, cortisol's low in the morning. I mean, that's true, but the body has to respond to stressors at a much faster rate than every 24 hours. There are stressors that you do. Every time you do a stressor, something is causing a upsurge in melatonin and cortisol. And I would argue very strongly, based on what I'm seeing so far, is that this is probably why we have a cortisol-rich environment, or that we are generating a cortisol-rich environment with all these stressors. How many times do you see a kid sitting there on his cell phone or whatever in the middle of a dark room, we're not getting enough exercise, we're not getting enough sunlight. And sunlight is something I'm starting to focus a lot on, because it has some really interesting effects in that there's a slight dip in melatonin, but then there's a long range increase in melatonin when you're out in the sun. So it's kind of like I believe melatonin is more of a protectant. And if you get too much sun, it gets depleted. You have to think of melatonin as a consumable. And then if over time, the body catches up and builds up more and more melatonin, and it keeps on going up. So all I'm saying is there is a baseline cortisol and melatonin effect, but the minute you introduce real life into it, there are all these transients that we have no clue where they're coming from yet.
Oh, no, that's fascinating. But I think it comes back to what we've talked about before, of how important the shade is, because it's dark, and then me and caves and going and getting some, like a bit of darkness just as a break from the light. So I think it's all pretty interesting. We're going to have to stop soon, because we've gone on a long time, and I think we've covered a lot of different topics. Is there anything that you wanted to end on? Because there's so much to say. We'd be here forever.
I think that's
because I don't want to give it all away. I think it's something that these are really interesting discussions. And I think a lot of this kind of information and this idea of optics, people need to absorb it slowly over time. And they always have to know, how is it relevant for me? And importantly, as you said, just to keep questioning things.
Yeah. I guess I would end it. One of the things that I think we need to look at is that how we measure things, what conditions they are, how we plot things, it all matters. I mean, this graph here is just a simple example. You look at, what I've just laid out is true, and that we want to have a lot of photons out in the 1500 to 1600 region, then, I'm never going to be able to measure that using a silicon the spectrometers that they got. It's not designed to be able to measure it.
That's exactly the problem with a lot of optics, including biophotons. Just because you can't measure something doesn't mean it's not there. Or you might be just using the wrong measuring device for that particular part of the spectrum.
Yeah, and I guess I just end on this is that in very simple terms, sunlight, when we're outdoors, we have in optical watts, we have equal amounts of near infrared and infrared relative to visible. You go into the trees, all of a sudden it absorbs some of the visible, reflects a lot of the near infrared, and it increases the ratio. You get to fire or incandescent bulbs, it's 10-1. All this stuff we're doing with the circadian or the color shifting, color tuning, even out to 1,000 nanometers, it's almost insignificant. And as far as what the mitochondria cares about, at least, and the rest of the body functions, I know you can convince people that it's better for or that it's doing all this color shifting is good because they can see it. But in reality, I don't think it's the most important thing we should be focused on, especially given the coincidence that we're having all these mitochondria or metabolic diseases, and we need to start understanding what the entire solar spectrum is and get kids outside. I'm sorry. They need to go outside. They need to get some fresh air. They need to run around. And until everything else is kind of, insignificant in a lot of ways, I think.
Exactly. And also if people need to use light inside for their jobs or whatever, we need to understand properly about indoor lighting.
Yeah, I mean, I think that Glenn just published his data on eye color contrast. In a matter of hours, he was seeing significant change in or degradation in color contrast under LEDs versus incandescent. This should have been done before we ever introduced LEDs. Now we've introduced LEDs, and now we've got to figure out how much do we have to add back in order to correct the problems being created by, a poorly thought out design.
But also you're completely right about ratios, that the ratio of light the sun pumps out throughout the day is different pretty much every second. So that's the other, how to factor it in, to change the ratios of indoor lighting based on morning, noon and evening to make it more like the sun.
Yeah, and then all I guess I'm just saying is that if we could find a way to appropriately measure, quantify some of these optical effects and what we're seeing, the effects we're seeing, it's all going to indicate that we probably need to introduce some kind of broadband emitter back into our lives. It can be as simple as, if we can get the government to get off our back and let incandescents come back, great. Glenn's only just taking an incandescent desk lamp and putting it next to a computer monitor and seeing these huge effects on color contrast in the eye. How many other processes are being negatively affected?
There's loads of them. I know other physicists, and they all have a different reason of why they dislike the artificial lights based on different effects on brain waves or focus, or there's all sorts of things. But I think it's like I said, I think this is still just what I'd call it science in its infancy.
I agree. Like I say, we're just trying to bring out that, there's no difference between, calling something heat means that you really don't understand what's going on, because it's all photons. It just is. And you happen to be at different energy levels. But the idea that the body isn't taking advantage of all the photons that are available in some way or another, even as you say with a tree, letting something else live, the understory, that type of stuff. There's an advantage to the tree if there's good underbrush, or whatever.
There's lots of things we could speculate and talk about for ages. But anyway, I'm going to have to stop now because you've given me way more of your time than I was expecting, which is great. But then we've only just got going. But I just want to thank you for your presentation. And there's constantly more work coming from you and Bob and Glenn anyway. So this is just the beginning as well.
I appreciate your time. I always enjoy the conversation.
Thank you very much for watching. I hope you found this interesting and informative. If you want to learn more about quantum biology, check out my brand new Quantum Starter Kit. So it's a beginner's course. It's very affordable. It's $19. It's a video recording with a slide presentation, but also you get the handouts as well, because some people like to read as well as listen or watch. So thank you for watching, and thank you for supporting the channel.